technical issues here. So hello, everyone. Thanks for joining us today on another APS seminar series. It is my pleasure to introduce Lucas Roberts. Lucas grew up on a family farm in central Illinois. He did his undergraduate at Iowa State University, double majoring in agronomy and genetics. Then he completed his master's degree in crop sciences from the University of Illinois, studying water use efficiency in maize. Currently, he's a second-year PhD student in the Applied Plant Sciences program in the Department of Agronomy and Plant Genetics. He's a member of the Lorenz Lab, developing soybeans for a winter oil seed intercropping system. He's also involved in the APS Grad Club and a co-chair for the Plant Sciences Symposium.
Okay, thank you, Maicon, for a second time around. Thank you for coming to my seminar today, which is titled Battle of the Oilseeds, where I will be talking about the characterization and genetic variation of soybean traits relevant to a soybean-pennycress intercropping system. This figure on the right shows how these two species are interacting. My outline for today will include a background on an introduction to intercropping, primer on pennycress within pennycress history, moving on to why soybeans are important, and the best of both worlds, how these two species can live and interact in this intercropping system, with later focus on my thesis chapters. To get us started, an introduction to intercropping. Intercropping has several key tenets. One of them is resource allocation, where resources, whether soil, nutrients, water, or even sunlight, are separated by time and space. Some of them separated by space include alternate rows of species. This here is an alfalfa kernza intercropping system, which is also separated by time, where the kernza is harvested before several of the alfalfa is harvested. On the right here shows a Kentucky bluegrass and maize intercropping system, where the cool season perennial grass can grow and collect sunlight before the maize shades it during the summer. Moving on to pennycress, a primer on pennycress. Pennycress is a member of the Brassicaceae family, so that means it has the potential for oilseed. These are a picture of the seeds. I want to highlight that they are smaller than a canola seed. They have this very characteristic thumbprint, the pattern of grooves and ridges that help in its identification. And on the right here, you can see a flowering pennycress with the fruit capsules, which are known as calyx. So the scientific name of pennycress is Philosophy arvens. As I said before, it's a member of the Brassicaceae family. This figure on the right here shows its relationship to Arabidopsis. So it is a second clade, a separate clade, and they are separate but similar. And I'll go into why that's important. Some of the names include stinkweed or field pennycress. If you have the pleasure of seeing a pennycress field, you will know in the springtime has a very pungent garlic-like odor. The genome size includes 7 chromosomes, and the genome size is around 500 megabases, around 5 times the size of Arabidopsis. It is native to Eurasia, but it has been naturalized throughout the US and North America for a while. It is typically a weedy species. Focusing on some of the chromosome aspects of pennycress, the top figure here shows an ancestral Brassicaceae, whereas pennycress has 7 chromosomes and Arabidopsis has 5. And I want to highlight the similarities. You can see similar chromosomes are colored the same between. And so while there is rearrangement, there's still a lot of synteny, which is a powerful tool that we can use, a powerful tool that pennycress breeders can use. And so how do we utilize Arabidopsis? A lot of the foundational mountain of work for Arabidopsis is directly translatable to pennycress. One example here is on a key domestication trait, so shattering versus non-shattering. In the Arabidopsis mutant, they found for non-shattering, the same mutation was found in pennycress. And so these tools really enable the rapid domestication of pennycress. A brief history on this crop, on this new novel crop. It was first reported for an oilseed potential in the 1940s, followed by the identification of uric acid. So uric acid is a major anti-nutritional compound that has been bred out of canola. And around the early 2000s, it was identified as a new source of fumigants, of insecticides, fungicides to replace methyl bromide. And I want to highlight this further, that this pennycress, this crop, has a really strong ability to suppress competition. Early in the 2000s, the USDA started funding research for both food and fuel purposes, with the first pennycress breeding program being started at Western Illinois University around 15 years ago. And just last year was the first on-farm farmers trials of some improved cultivars. So the picture on the right here shows a golden seed. Wild type is a black seed. And most of this research I'll be talking about today is funded by IPREFER, or Integrated Pennycress Research Enabling Farm and Energy Resilience. Going back to the genetic resources. So I said that pennycress was similar to Arabidopsis, but it actually has a very large percent of repetitive elements, which compose of multiple TE families, similar to other genomes like maize, which has a large TE content. Looking at the genetic diversity of the population structure, the figure on the top right shows that there is some population structure. Those triangles are in Armenia, or European germplasm, but most of the US germplasm really condenses in one direction, which only explains around 10% of the variation. This picture on the bottom right shows a genetically transformed pennycress using GFP, green fluorescent protein, and the methods that Arabidopsis is transformed via a floral dip work the same in pennycress. Moving on to the benefits of pennycress. So why do we want to plant this cover crop, this cash cover crop? Well, one of the primary benefits include ecosystem services. So this supports pollinator health. Early in the springtime, these fields are full of flowers at a time where few other plants are blooming. In addition, it supports weed suppression. So pennycress has the ability via releasing chemicals in its roots to suppress germination in particular, but also shading out of competing species. As I said before, it's a cash cover crop. So you have the benefits of a cover crop with ecosystem services, but you can also sell the seed for oil or food purposes. In addition to cover crops, they also have the ability to absorb excess nitrates in the soil. So that prevents their leaching into the soil, groundwater, and to the lakes of Minnesota. It has the ability to extend the growing season. So pennycress is a winter annual. It is grown, it germinates in the fall, it forms a rosette over the winter, and it bolts early in spring. So this maximizes the number of months we can have crops in the field. And lastly, it increases energy security. So this is an alternative oil seed. We are reducing our dependencies on canola or soybeans as our only oil seed crops. So why do we care about pennycress? What are the uses of it? Well, the seed contains a fair amount of oil, around 36%, with the major fatty acid in wild type being uric acid. A yield would be on average 40 bushels per acre. And the fatty acid profile, so the ratio of the hydrocarbons, is particularly ideal for jet biofuel, aviation fuel, and for jet planes, as well as the seed oil has a potential for feed sources if some of these anti-nutritional compounds are removed. So why soybeans? So I've told you before that pennycress is a winter annual, and it does most of its growing season in the fall. So on the top here, you can see it germinates in the springtime with an early senescence in May. This is depicted in a linear fashion on the bottom. You might be familiar with other winter oilseeds such as winter camelina. Winter camelina lasts into the summer around a month longer. And so you might say, what if we intercropped it with corn? Corn is typically planted earlier than soybeans in the upper Midwest, as well as corn cannot really tolerate stress the same way that soybeans do. We say soybeans have a greater amount of phenotypic plasticity. And so these two species work well on a time frame together. And when you plot biomass, they're only interacting around 6 to 8 weeks down here. And it is during a point where the soybeans are still in a juvenile state. So taking a step back about the importance of soybeans. Soybeans are a major oil crop, but they're also a major protein crop. Around 80% of the seed is included in meal. If you break that up, around 30% of that meal is protein. This is a major source for animal feed, both domestic and globally, as well as some soybean protein food products. The remaining 20% is oil, with the majority of that being food-grade oil, but there is the potential for biodiesel. And this oil can also be converted to other polymers for more industrial uses. Taking a step back, looking at the global level, soybeans abroad and at home. The US is a major producer of soybeans. This figure is a little bit outdated. As of last year, Brazil and the United States are around on par, about both 35%, with other South American countries like Argentina comprising a significant portion. When we narrow down onto the United States, soybeans are grown on a large north and south gradient. And so that corresponds to different maturity groups in soybeans. Illinois is the leading state in the US, followed by Iowa and Minnesota. So Minnesota is has the third most production total. Moving on to soybean diversity. So a lot of you might think soybeans are just this yellow seeded crop that's planted in the fields. I want to highlight that there is a rich history of domestication and a rich diversity of germplasms available in the US germplasm collection. This is just a picture of different soybean seed colors. This applies to a plethora of traits. We're looking at branching angles or what have you. On the right here shows a phylogenetic tree of some soybean diversity. So I chose this figure in particular because I'll be using this diversity panel in one of my later thesis chapters. I want to highlight the different countries of origin with this light blue cyan color being United States. This figure looks at the wild relative of glycine soda, and I will only be looking at the elite and landrace cultivars. So where do soybeans fit in with pennycress? So one of the ideas is to increase yield on a per acre basis. This figure in the right column shows soybean and pennycress when grown together in an intercropping system, they yield more than soybeans alone. I also want to highlight that in this system, soybean yield is decreased. And so one of our long-term goals is to develop new soybean varieties that minimize this yield penalty when the two crops are growing together. That is a long-term goal. It takes a breeding pipeline 9 years or so to go to cycle. And so some of the short-term goals that are achievable in my time here at Minnesota include utilizing breeding lines to look at and characterize this genotype by cropping system interaction to see if it exists and ways to maximize this interaction, as well as identify traits that might be important for adapting soybeans, looking at a trait-involved approach towards breeding. Traits that can be particularly selected in a monoculture and a traditional breeding program. This is a figure of a recent publication that just got accepted of mine, looking at breeding for intercropping systems. So this will be our roadmap for today, and I'm going to take you into the different quadrants. This is the source for that. So focusing at the beginning, we first have to define the problem space. What could be some constraints? Is there a need for this? Can we solve these through more agronomic processes, or do we need to breed and develop new varieties that have increased tolerances? And then to finally access the variation. Is there variation for a trace of interest? Can we realistically improve these varieties? Moving on to actually designing the breeding scheme, we need to see if we can test and select and breed for varieties in an intercropping system, or if we need to breed for it in a monoculture breeding pipeline. And so some of the things I want to highlight here in the intercropping breeding world, you can have varieties that have a general mixing ability or a specific mixing ability. So a general mixing ability would be one soybean variety that can work for 40 different pennycress successions, whereas the opposite would be a specific mixing ability. I need to combine this soybean genotype with this pennycress variety. Moving on to how would you actually select for this process. So as I said before, depending on your traits, depending on your logistics, you can select for crops separately in a monoculture. You can select for them somewhat limited in both, or you can co-select crops, i.e. you're growing these soybeans next to pennycress and making the direct selections there. So ultimately, you have to do final yield trials in an intercropping system, and that is how you would release varieties specifically adapted for intercropping. And like with many breeding pipelines, they are recycled. This is a cycle. The genotypes are recycled for further improvements in the future. So moving into a trait-informed approach, what are the idiotypes for soybeans? And this would also differ for whether the pennycress is intercropped or monocrop. And so it's looking at the top row here for a pennycress. Some of the agronomic traits that I am considering would be reduced shattering. If the plants are shattering, those seeds are germinating and could cause stress later in the season for the soybeans. I want to push that window earlier for early maturing to reduce that number of time that they live together. The architecture, I want to reduce tillers to let more sunlight through. And for abiotic and biotic, I want to reduce the stress that this pennycress applies to the soybean. Looking at intercropping soybean, I want plants that are more hardy, that are more tolerant. Some of these stresses include cold tolerance. You have to plant the soybeans a little earlier than recommended before the pennycress gets too tall, as well as reduced etiolation, so this shading stress tolerance. I want these soybeans to close the canopy faster in a later time frame than what is normal in a monoculture environment, as well as the other stresses such as drought tolerance or allelopathy tolerance. And the pennycress has the potential to increase some diseases and pests. So pennycress is a known soybean cyst nematode host, but it looks like that is not a major issue. Other potentials could be white mold is able to infect pennycress. Okay, so now that I've covered the background to help you understand my thesis chapters, we're going to move into what my thesis will be, starting off with chapter 1. So the main hypothesis, main question for chapter 1 is, are some soybeans already adapted for intercropping? And the way I will determine this is I will test different soybean genotypes and look at their growth, their development, and yield under pennycress. So this project looks at 40 different soybean sessions. These include commercial varieties, breeding lines, and PI accessions. These plant introductions, these national germplasm-based, more landrace type cultivars. And I want to highlight that some of these breeding lines in trial were developed by the previous soybean breeder, Jim Orff, specifically bred for organic systems. So these varieties might be high stress tolerance, they might close their canopy faster to have a more competitive element. Moving on to the experimental design of my first chapter, the pennycress will be planted on 30-inch rows, and the soybeans will be planted on 30-inch rows in a gap row between 3 rows of pennycress. This enables a short window for the soybeans to get established before the canopy closes. So you can see on the right here, as the pennycress senesces, this is at pennycress senescence, these soybeans are in full shape. This is a stressful environment. You can imagine this large amount of pennycress biomass is pulling a lot of the moisture out of the soil. This is a bird's eye view. So our lab utilizes UAVs, unmanned aerial vehicles. And this is the same field in 2020 in June versus August. I am using a split block design where I have 3 blocks of my 40 genotypes planted, interplanted with pennycress, and 3 of monoculture. And I want to highlight on the right here, by August, for the most part, these genotypes are closing the canopy. You see there is some less row space in a monoculture. I also want to highlight the genetic diversity. So you can see all the different shades of green corresponds to the varieties that I'm utilizing. In using UAV systems, 2 of the traits that I'm looking at include vegetation cover. So this is that canopy coverage. Early on in the season, I want to highlight that a soybean monoculture, soybeans alone, close their canopy faster than a soybean intercropping, but later in the season, they meet up. Also, we look at a mean green leaf index, so the greenness of the score, and there are no significant differences for the greenness. So in a field setting, these plants are not showing chlorotic leaves. Moving on to the agronomic trace that I'm looking at, one of the most important being yield. On average, yield has decreased around 5 bushels per acre. This figure on the right is over 2 years in 2 locations of my 40 genotypes. And you can see the blue line is a 1-to-1 line. So on average, for all my 4 different genotypes, their yield in monoculture is greater than the yield in intercropping. On the far right is a commercial variety that performs very well in monoculture, but does rather mediocre in intercropping. On the bottom left is a variety called Mingold. This is a field marker that we use that is a photosynthetic mutant, so we would expect this to have low yields. And I want to highlight this, another industry variety, this BS line on the top. This shows great promise. It yields above everybody else in intercropping and is rather close to that 1-to-1 line. And so you'll see this line later in my presentation as we are focusing in on why this is occurring. There is a plethora of other traits that I look at. This is just a list of seed quality traits. So the samples from this experiment, the seeds are run through an NIR, and there's an estimate on the different types of oils, the acids, as well as amino acids. And there are no significant differences among these traits. So the seed quality is not decreased. And I just want to highlight a few of the traits that I skipped. So lodging is generally not affected. Height under Pennycress is around 5 inches shorter. No significant differences in protein or oil, just slight changes, as well as the internodes are longer. So there is a shading stress and etiolation occurring. Moving on to the image analysis. So I'm taking 6 plants per plot and looking at the structure, the architecture of the plant. This is my setup I have in the seed house where I image the plants. And so some of the things that you can get with imaging include internode number as well as length. And so this picture on the left shows very young soybean seedlings. On the left is in monoculture, on the right is in intercropping. You can see this etiolated hypocotyl, this shading response is occurring. On the right, you can see the pods, but as well as the branching differences. So what is interesting in this system, this is the same genotype. On top is monoculture, on the bottom is intercrop. Under intercrop, the stress-induced branching. So this is an ideal response. The more branches there are, the more nodes there are. The more nodes there are, the more places for pods, the greater the sink potential is. And this is not seen across all varieties, but it is a trend that we are observing. Moving on to the second objective of my chapter, looking at, okay, so if these soybeans are adapted for intercropping, if not, to see if it's possible to select for new varieties, to develop, to breed for them in a conventional program. So I want to highlight some of the challenges it would be to breed in an intercropping field like those last pictures showed. So one of the major factors we are considering is necessitated by the reduction of field space. So here at the university, the pennycress is still semi-shattering, and those seeds live in the soil seed bank for a number of years, and they're actually running out of field to plant new pennycress research fields on, as well as some traits that are able to be screened in a monoculture environment, like height and canopy coverage, as well as some that you cannot screen in a conventional program, like allelopathy tolerance or shading tolerance. So looking at the correlation of soybean traits, just to remind you, I'm only looking at soybean traits. These are soybeans under a monoculture and soybeans under intercropping. And I want to highlight, so taking a step back, you can see general trends are the same, but I want to highlight 2 differences. So the correlation between maturity and yield in a regular monoculture field is 0.6, whereas in intercropping is 0.7. So this means a later maturing, a larger maturity number is correlated with greater yield. For the correlation between yield and height, it's around 0.7 under monoculture and 0.9 under intercropping. So this would indicate under an intercropping system, you would want later maturing and taller plants. These are 2 things that are strongly selected against in a monoculture field. I want a shorter plant that does not lodge, and I want an early maturing that does not have the risk of being frosted. So these further highlight the need to have separate breeding targets and separate breeding pipelines. Looking at the heritability of these traits, so can we select in an intercropping system? 4 of my main traits here include maturity, height, lodging, and yield, and this is a measurement of heritability. This heritability measurement is also known as reliability. It's calculated over 2 years in 2 locations. For some traits, such as age and maturity, the reliability is very similar for maturity, but for other major agronomic traits, the heritability is much lower in these systems, indicating this is a more stressful environment. There are more factors going on that there's more variation that allows poor estimates of heritability, further implying the need to select varieties in a monoculture breeding trial. Okay, as a final thought for chapter 1, looking back, I want to highlight that this is all relative to the environment. As several of you know, last year was a very severe drought in Minnesota. So for 2021, my yields looked very good, but last year, the soybean saw a lot of stress. On the right, I want to highlight, this is the board of the blocks where pennycress on the left and soybeans on the right. You can see much lower biomass on the left because the pennycress just sucked a lot of the water out of the soil profile. Here on the left, it shows that late in the season, on a genotype specific interaction, some genotypes were not able to close the canopy. And this is just the volunteer pennycress, the shattered pennycress that is further sucking moisture out of this cropping system. So these are some of the challenges that, yes, there is great potential, but it is also limited on the environment. Transitioning to chapter 2, how does intercropping affect soybean physiology? The first objective is to develop a screening method for assaying allelopathy tolerance. So like I said before, we can't really select in the field, so I need to select in a controlled environment to screen a number of genotypes, much greater than just the 40 that we studied before. So can we replicate this cropping stress? And then once we have it replicated, are we able to induce this? Are we able to screen for them? So going forward, I'll describe how I have some greenhouse and growth chamber pilot studies where we were able to apply this stress, as well as discriminate some soybean responses on a genotype level. I want to focus you back into the roadmap today. And so this is along the lines of identifying traits of interest observed in a monoculture environment, in a monoculture controlled environment. So this is some pictures from the growth chamber experiment. I have done a number of them. And here, just to give you a little background on experimental design, different tags will denote different percentages of pennycress powder intermixed, seed meal intermixed into the soil. On the right is some joys of working with growth chambers. So this is a picture over the summer when my growth chamber reached 50 degrees centigrade, and I had to restart it. So I want to take this time to ask my audience in the room in person, have you heard of allelopathy before? Are you familiar with it? Okay, so I'm going to describe it a little bit for everyone else. So there are different forms allelopathy can take, some active, some passive. And the more active roles, plants can exude, can release chemicals, molecules in their roots that will actively suppress germination, suppress plant growth around them. They can also actively release volatile compounds. So I want to remind you that pennycress has these volatile compounds. If you will go by a field, there's a strong garlic scent. When I open my growth chamber doors, I'm hit in the face with a wave of pungent smell. But allelopathy can also work in more passive ways. When plants are dead and as they decompose, some of these chemicals released have deleterious effects on plant growth. And one of the studies previously published showed that winter oilseeds had this phytotoxic, had this allelopathic effects, and in particular in pennycress, we're able to suppress weeds up to 100%. And I can attest to this. In a pennycress field, while the pennycress is alive, there is virtually no weeds. This effect breaks down as the pennycress is harvested and removed, though. Focusing down to allelopathy in pennycress. So typically in Brassicaceae, allelopathy is present in the form of glucosinolates. Pennycress only has one form called sinigrin. In other cruciferous vegetables, sinigrin has a number of well-characterized effects. And I want to highlight here the three in red. It will affect, reduce, suppress antibacterial, antifungal, and biofumigation. So I want to remind you all that soybeans have a symbiotic relationship with bacteria in their root nodules to fix nitrogen. So there is the potential that this allelopathic chemical could be reducing nitrogen fixation in the roots. Also, there is some debate in the literature still if synegrin is the main allelopathic compound or if it's direct decomposition of alloisocyanate, also known as mustard oil, is responsible for the majority of these deleterious effects. Moving on to pennycress and soybeans. For research done at this department has shown that biomass under winter oilseed, whether winter camelina or pennycress of soybeans, was reduced. So there is this effect going on. There's this deleterious reduction in biomass when soybeans are intercropped with these winter annuals. So to get us back on track, this chapter is about developing an assay. So can we look, can we measure, can we record genotypic specific responses? And so this figure shows my 40 genotypes from the summer field, and I chose 2 of the lowest performing and 2 of the highest performing lines. And I want to keep in mind my y-axis here is change in average bushels. So this is comparing monoculture to intercrop. So these varieties were reduced by 20 bushels, where some varieties in 2020 saw slight reductions. This is just a picture of the pennycress seed meal that was used in this study. This is whole pennycress seed that is ground up to a fine powder. And then it is mixed at different percentages into the soil. My genotype's not showing up here, but I have 4 different genotypes as the previous slide showed. And I want to highlight that you can see differences. Even in a controlled setting, some genotypes have larger biomass than others. So it's always important to compare to the control. Some general effects that I want to highlight include this yellowing. So these plants are chlorotic. Maybe they are experiencing their nitrogen symbiosis reduction. I also want to highlight genotypic specific effects. So you can see the genotype in the middle picture on the far right column shows great reduction in leaves, shows harm, where some varieties appear to be more tolerant. So I'm looking at this data, analyzing it through an analysis of variance, looking at the different amounts of pennycress percentage on the weight by volume in the soil. The 2% was significant. And so moving forward, that will be the amount that we will use. Looking at a genotype specific level, I have my 4 genotypes. I have a reminder in the top right here of their putative, their assumed susceptibility resistance. 3 of these varieties showed susceptibility, where my BS line showed the most resistance. And I want to remind folks, this is a 1-to-1 comparison. So if it was a 1-to-1 level, that means under the control versus pennycress treatment, they would be the same. So all genotypes saw a reduction in biomass, but 1 saw less of a reduction. With any good experiments, it's imperative to have a positive control. And so there is a plethora of pennycress research done here at the University of Minnesota, and they have identified mutants that actually have no glucosinolate in them. This is a glucosinolate null mutant. And so I performed the same type of study over different soil percentages using ground up pennycress seed that does not contain any glucosinolates, any synegrin. And there were no significances among the treatments, further supporting the idea that synegrin is responsible for these deleterious effects. And I want to highlight some of these varieties actually perform better under this glucosinolate null. And so that could potentially be, well, there are other nutrients, there's carbon and nitrogen in these seeds that could be acting in a beneficial way. Moving on to objective 2 of chapter 2. Once I have this assay working, there's a need to screen diverse germplasm and elite germplasm for allelopathy tolerance and shading tolerance, which is taking a back burner in my research. So there's 2 different panels that I'm going to utilize, that I'm going to screen. This first one here is a panel of 429 diverse accessions. This is that phylogenetic tree that I showed you before. I am using a subset of just the elite breeding material and elite land races, which has publicly available resequencing data. And the second panel is one yet to be made as a human breeding panel. So to give you a little context, up in Minnesota, we're at one of the more northern latitudes for growing soybeans. And so we cannot grow a lot of these diverse materials in a Minnesota field. So there is a need to develop a diverse panel for Minnesota's setting that can be used for a plethora of other traits in the future. These are a picture of some of the diverse lines grown when I transitioned from a growth chamber to a greenhouse study. And I want to highlight some of the stresses that are being observed. So this picture on the left shows a delayed growth, a reduced vigor. The picture in the middle here shows a potential apical damage. So I would like to imagine that the pennycress synegrin in the soil is being translocated through the plant, is damaging that meristem, and is inducing multi-branching, but I cannot prove that at this time, as well as just general stunting. So there is some misformed growth occurring here. Leaves are not the right shape, biomass, growth and develop is not occurring as it should. Looking at some of these results across diverse genotypes, I want to highlight just how much this pennycress seed meal can reduce biomass. So in a control setting, the average biomass is around 8 grams, and it reduces all the way down to 2 grams. And so I want to highlight that there were genotypic specific effects that were significant in an analysis of variance, looking at across these 8 different genotypes. So as a conclusion to my chapter 2, I have several questions I'd like to open up to the audience at the end of this presentation. So pennycress seeds also contain tannins, not just glucosinolates, so there could be other compounds in the seed meal that are concurring these deleterious effects. One of the solutions would be to remove these confounding factors by just using synthetic synegrin. One of the problems is the price tag on that. It's around $240 for 100 milligrams. Even if I were able to afford that, I do not know the exact percentages that pennycress is releasing synegrin into the soil to replicate this in a more controlled manner. Pennycress advancement has been rapid. There's been a lot of improved lines. These golden seeded lines I told you about earlier in the presentation. I'm not working with the newest lines yet. This research has focused on one of the earliest lines called MN106, the non-shatter variety. And lastly, as I alluded to before, I would really like to develop a method that really proves that this synegrin is translocating between pennycress and into soybeans. So one potential method for that could be radio labeling. You could have carbon-14 labeling in the pennycress, and if that was detected in a nearby soybean, you could tell that for sure, yes, this is occurring. I would like to think it is occurring, but it is not nailed down at the moment. Moving on to chapter 3, what genetic factors might underlie intercropping tolerance? So the first objective for this chapter would include running a GWAS, or genome-wide association study, on the previously described 429 diversity panel. And the goal of this is to discover quantitative trait loci that are linked to controlling variation for putative allelopathy tolerance. So going back to our roadmap, this is where we are in the bottom right, selecting for intercropping. So we are selecting in a monocrop system, we are selecting outside of this intercropping environment. And so what does a GWAS look like? These figures have the different chromosomes on the y-axis, 20 in soybeans, and their significance of how well each marker, each SNP, each single nucleotide polymorphism is linked for your trait of interest. Anything above this green line is considered significant. This figure is just made on an alternate trait I'm looking at for a side project. But going back to the power of GWAS, what can it do for you? So for this project, it has the potential to characterize and identify genetic architecture underlying allelopathy tolerance. As I said before, we test each marker. There are 479,000 in this data set that I'm looking at. And it tests their linkage with your trait of interest. This is ideal for complex polygenetic traits. So I hope I described to you before all the different ways that soybeans can respond to this trait, and putatively, theoretically, it would have a complex polygenetic cause. And lastly, what do we do if we find a significant marker? Some of the long-term goals would be to validate that SNP with the potential for marker-assisted selection in a breeding program to actually breed varieties for this intercropping system. And I want to highlight that is 7 to 9 years, well beyond the timeline for my time here at Minnesota. Moving on to the second objective is to look at how this intercropping stress affects gene expression. And so the way you do this is an RNA-Seq or RNA sequence experiment. And I have plans to look at several different seedling tissue types, so root, cotyledon, and true leaves, looking at 1 or 2 soybean genotypes grown in allelopathy stress followed by a controlled environment. And one of the ultimate goals of this objective is to look at how the gene expression, so how genes are either over or under expressed because of this intercropping stress, in a gene ontology terms. So I want to look at assign a biological significance to group and cluster these gene families and to attempt to explain what is occurring. And then finally, you can compare these results to putative GWAS results. And if they align, that would be a home run. So to give you a little bit of background on what Rna-Seq is, as I said before, I would use a small number of genotypes. You would have 2 different treatments, 2 different stresses. One would be a controlled environment, one would be under a Pennycress stress environment. And this would reveal the presence as well as quantity of RNA. And it's at a given snapshot, a given second when you were collecting that tissue. And this can identify genes that are overexpressed or underexpressed relative to the environment. And the ultimate goal is to assign a functional annotation. So what genes, what families, what groups of genes are overexpressed and underexpressed together, also called GO terms or gene ontology terms. And this subsequent clustering can inform biological significance of what is actually occurring in the plant. Moving on to future objectives, as I said before, there is a need to develop a breeding panel. I have plans to develop a core 300 genotype panel and a smaller mini core of around 100 genotypes. These genotypes will need to be, these accessions will need to be genotyped via resequencing. There are plans to perform a clustering analysis to maximize diversity relevant to Minnesota. These are 2 different publications of core hunters or genocores that I'm looking into developing this panel. I am getting my feet wet into looking at the population structure of how I will eventually do this to maximize diversity. This is just looking at chromosome 1 of soybeans from the publicly available SNP data set. And I'm also looking at different methods of clustering. So as I said before, to form 100 or 300 core genotypes and have them cluster in 3-dimensional space off of a principal component analysis. Lastly, I want to take a step back and remind you of what my title was today. So my title was Battle of the Oilseeds. So I want to put a more positive spin on this, that battle can have some positive connotations. It can include a lengthy engagement and a counter and competition. So this here is to illustrate that there is no bad guys. Pennycress and soybeans are both good guys working together, that this interaction is important, that there are a lot of potential benefits, and that through breeding and development, a solution should be possible. Lastly, I need to acknowledge the Lorenz Lab. Aaron Lorenz is my advisor, my advisory committee, including the soybean breeding technicians, postdocs and graduate students, visiting scholars, undergrads, as well as I prefer collaborators. As I think you can tell, there is a whole lot of data collection in this project, something that I could absolutely not do by myself. And I'm very grateful for all the help that all of these names up here have given to me. And with that, I would like to open the floor for questions. Thank you. If someone can manage the chat questions for me. Okay. I do not see any questions in the chat at this time, so I'd like to open the floor to the students that are here in person.
I have a question.
Yep.
So in Minnesota, most soybeans are rotated with corn. Right?
Correct.
1 year you can plant soybeans, the other year you plant corn. But in pennycress is a perennial, right?
No, pennycress is annual, a
Oh, it's
winter annual.
Okay. Okay. So my question is, for example, if you have remaining seeds that you harvest, they feather, they remain in the field. What would be one way to manage, because they are both, like, they are not from the same family, but could the pennycress be a problem for corn production? Like in terms of, it could be considered as a weed, and then in terms of like weed management or something like that.
Yeah. So I'm going to echo the question that was asked. Could pennycress become a potential weed for corn systems as the seeds persist in the soil? I'm going to say that there is a weed scientist on the grant that is funding me that is looking into that. I'm also going to say that pennycress is not that competitive in a corn environment. So pennycress is dead, it senesces by early summer, and the corn plants for the most time are more vigorous, you can outshade it. I also want to highlight that, so I talked about a corn intercropping system. There are other collaborators on this grant that are looking at planting, so the rotation, ideal rotation would be corn, pennycress, and then soybean. So they're looking at corn, soybean genotypes that can tolerate a fall planting of pennycress and not kill the pennycress. Yeah. Further options include pennycress as a broadleaf and corn as a grass. So that's a clear separation of potential herbicides that you could use to control this species. Is this the first question? Okay, so Nicole asked, are you differentiating between true allelopathy and other competition suppression mechanisms in your trait? Yeah, so as I said before, I would like to think that this is true allelopathy going on, but I would like some more concrete proof, be like a radioisotope label to prove that this is occurring. I also want to highlight this competition and suppression mechanism. So in the field environment, once a pennycress has been harvested, its allelopathic effects are no longer, and this intercropping system tends to be very weedy. So weed management is an important component for later in the season soybean growing. Yeah, great question.
Is this cropping system where is it all still underdeveloped?
Yep. Yep. So Amanda asked, what is the level of actual growing pennycress? So last summer was the very first summer of on-farmer trials. So there's been a lot of research at the university level, and this is run by our collaborators at I Prefer and Covercress out of St. Louis. And so I believe all the trials were in Illinois last year. And so this is a brand new novel crop that is, there's still a lot of work to go before it is feasible for the farmers.
And yeah, those were on-farm trials, but you said the 2021.
Correct. So like I have on-farm trials, but we're having on-farmer trials to get the farmers involved, to get the producers involved, to get their inputs and their concerns addressed. Yep. Cool. No, I think that's.
Oh, that's the same.
Yeah. Are there any other questions? Yes.
I don't know if I missed it, but what do you consider is the most like challenging part of integrating this crop into the crop?
Yeah, so there's a lot of benefits of intercropping with pennycress, as I described before, but there is also a lot of challenges, a lot of hurdles to be overcome before this is ready for a farm environment. Some of those including the stress pennycress applies onto soybeans, but also on a more practical agronomic approach. I alluded to before, this pennycress system with pennycress dies, the field becomes very weedy. And so I am out there weeding, hand weeding 3 times a summer. Is that practical for farmers at this time? No, but there is work done on the more weed science herbicide side of things for getting applications that you can spray once soybeans have begun flowering. So that is a big challenge for me logistically and farmers in the future.
I'm not
Yeah.
sure what slide, but you showed the one-to-one.
Yeah. Yep. So this is over 2 years in 2 locations. So I did not specify. My research locations include Rosemount, Minnesota, and Morris, Minnesota. Oh, thank you. Okay. Does planting pennycress at different densities change the responses from the soybean? Or does the persistence of pennycress seed in the seed bank mean that you wind up with a certain density after a number of seasons anyway? Yep, so that's a great question. I am not on the pennycress side of this research. There are other professors in this department, particularly Samantha Wells, that might be able to answer this question. I believe you're looking at different densities. I also want to highlight that it is dependent on the variety of pennycress that you grow. Some varieties are tillering and some are non-tillering. The ultimate goal would that they would not persist in the seed bank. Right now, there's still a tendency to be a weedy on the weedier side, a weedier species. As I said before, this would be a winter annual. This would be something that would be planted every fall that you wanted to grow. And if any volunteers occurred, those would be treated as weeds and removed, just like volunteer corn is removed in a soybean field.
Is that related to yield at all, or is that just related to?
Yeah, so that's a great question. There were slight differences, but as I said before, not significantly different. I can tell you a little bit of background. So one of the major breeding focuses of soybeans is that they increase protein. Proteins classically have a very strong negative correlation with yield. So I could get yield by having bigger seeds, but bigger seeds would have less protein inside. Does that answer your question?
That was helpful, yeah.
Okay.
Yeah.
In the context, yeah. So looking at the quality, making sure that this intercropping stress does not reduce the quality of the soybeans that the end goal end producer hopes to sell and market. Seeing no more questions, I think we're going to end this public seminar. I'd like to thank you all for attending. And for the APS students on the seminar, we're going to keep on this once we give everybody a chance to log off for the rest of this class. So with that, thank you.
Well, I've got a few questions for you, Lucas.
Okay.
Which I wasn't going to ask in front of everybody, but can you tell me why you switched from the growth chamber to the greenhouse?
Yeah, great question. So I need to scale this up. I need to screen 429 accessions.
Got it.
In a growth chamber, I can only screen 4 accessions at a time. And so there's a need to still keep some similarity. I don't want to run 20 growth chambers. I would like to do 2 replicates of this 400 genotypes spread out on over 2 or 3 greenhouse runs.
And did you change the media that you're growing the plants in from growth chamber to greenhouse?
I did not know. It's a soilless media down there.
And so I have one more, I'm the greenhouse person.
Yeah, no, that's what.
So in chapter 3, you're talking about your Rna-Seq data.
Yep.
And you were talking about looking at like growing them with a control and growing them with 2% pennycress, whatever.
Yeah.
And you talked about a stress response. And the first thing I thought of, because I used to grow soybeans long ago in the greenhouse and they got tripped like crazy.
Yep.
So how do you make sure that you're only really looking at that particular
Yeah.
change versus all the other wonderful things that happen in the greenhouse, like
That-
trips and mites and
That's a great question. So RNA-seq experiments are very particular. I need to make sure that these plants are not experiencing drought stress, because that will show up and could pollute it. And so one of the good things about this is I'm working on juvenile stress tolerance. So juvenile soybeans, these soybeans are going for a short time. I would almost be more inclined to grow the RNA-seq experiment in a growth chamber, which provides a little more control. Like you said, no pests involved, as long as the growth chamber doesn't go down.
Well, that occasionally happens, but the greenhouse can also get really hot.
Yeah.
So one last thing, and I feel like I'm now like dominating this, but Adrian Hegeman in our department
Yeah.
labels plants as C13.
I'm hoping you listen today.
Well, you might want to just reach out to him because it's a lot less. I mean, having done C14 stuff, you really would rather do C13.
Okay.
And he's got a C13 chamber, so I would make an appointment with Dr. Hegeman.
I'm glad you said that. Thank you. I need to do that.
Who's, I mean, everything is grown in
Not necessarily, thrips get into one part of your greenhouse and it's not over here and.
It could be separated by
and
space.
Thrips get up, thrips get down into the growing points of the plant, particularly soybeans. They just seem to really like them.
I will say for RNA-seq, you have a very small number of technical replicates or a very small number of genotypes. So ideally, you wouldn't see any spatial variation, but it is a concern.
If I were you, I grow in the growth chamber. Who does?
Here's my question. I'm just curious, how do you evaluate watching?
So in the soybean breeding program, we have a scale from 0 to 5, literally over how much the plants are falling over. It's at the end of the season, right before it is harvested. So I said I'm working with some PIX sessions. These diverse materials really tend to lodge, that they have too much biomass, the plants get too tall, the spacing between pods is too much, and it's an important consideration. Say again?
When plants etiolate?
When they etiolate, yeah. So I'm glad you brought that up. I saw etiolation responses, but they did not have any differences in logic. So that's a surprise, a beneficial surprise. Maybe because these plants are generally shorter, the taller a plant, the more likely it is to lodge.
So, for example, last year was very dry. How was it 2020 and 2020?
2020 was ideal. This is what I would say in an ideal situation. These plants are healthy, their biomass were visually almost the same. Last year was just so stressful.
Yeah, I mean, because when very dry year, you wouldn't have lodging problems, right? Probably.
Yeah, they got much shorter.
Or the wet year, you could have more lodging problems because of the
You were a soybean farmer. Like, how excited or not excited would you be about this kind of crop?
I'm excited because this is a cash cover crop. This has a second opportunity of income for me. Say the soybeans experience a dry year and the yields are reduced. Well, it would still have that pennycress yield to back up on. A lot of these benefits are good, but they're not a monetary incentive yet. There's current cutting edge work looking at providing monetary trading carbon credits. So maybe that could be monetary in the future. And lastly, as a soybean farmer, I would say this needs 5 or 10 more years of research before I'd be willing to risk it.
How do you harvest the pennycress?
Okay. No, that's a good question.
How do you
So-
harvest the pennycress seed if your soybeans are coming up?
The pennycress is harvested, I think it senesces in early June, late May, early June. The soybean plants are small at that time. And so one of my traits that I set up there was I want them not to be etiolated. If the plants are too tall, then the combine has a tendency to come along and chop off the tops. I also think I said in that trait slide, I want the pennycress to almost be a little taller, or at least have their seleaks be higher up, right? Increase seleak height. Because if they're harvesting,
You actually want your inflorescence tall.
I want them separated
You
by
don't really
space.
care if your sleeks are tall. You really want your inflorescence to be tall.
Where the
That's your
sleeks
sleeks
start on the stem, I guess is what I'm.
Yeah.
Not total height, where
Right.
those
It
sleeks
says
start.
increase sleek height. I don't really think that.
First sleek height, I want it to be taller up.
So,
Yeah.
I have lots of questions.
That's so-
So do I, I'm ready for input.
So what's the comparison with the RNA-Seq experiment.
What is
What are you
the,
actually gonna compare? Compare to what? Treatment to non-treatment?
Yeah.
Okay, and so you're gonna get what, 2,000 genes that are differentially expressed.
a large expression
So
and
then, so you think that because one of those 2,000 genes kind of
not one,
coincident with
the genotypology families could potentially putatively offer a biological explanation.
no, but I'm getting at
Yeah.
your comment about GWAS. So your
Oh.
comment about GWAS was
Oh,
that you're gonna find a gene or more than one gene that are coincident with a GWAS hit.
would be
So if you
significant.
got thousands of genes, what are the chances of finding something that might be coincident?
Oh, yep. So
It's like, it's
in
kinda high,
doing
isn't it?
and doing multiple testing, you have to do multiple testing corrections. And so having a higher significance, looking at all these different SNPs markers, even looking at all these different genes, they really have to be significant.
I guess there's a couple of questions there. So one is, you know, the LP in the GWAS population, so you know whether or not it's gonna break down with the gene or it's gonna be NKB or megabase or what. And so that will help determine whether or not you can do with this associated your a hit or a highly expressed or lowly expressed gene. in
If
the
it's
RNA
close
seq,
enough.
and if it's actually, you even have a chance of finding something that might be that is,
That is a great question. I don't know off the top of my head. I think it decays okay, relatively well, not as good as corn. But in running that GWAS, it's one of the outputs on GAPIT, and so I could pull up the LD graph if you wanted.
Just to get a sense of whether or not
How
that's
quick.
being realistic.
Okay,
Because if it's more than like 100 kb, probably not.
I think that's a very good point.
If it's like within a gene or two. maybe. So, right, looking at this, so how do you press that pillars?
Oh, yeah.
Are they branches?
No, there's full tillers. In the paper,
Pillars that branches that they branch from the base of the plant?
Mhm.
They do, okay.
I could pull up a figure. If you,
I just wanted, I had no idea, I just thought maybe I was typo.
So-
Maybe I've never really seen a thing.
I have plants in my office. I could bring in a pennycress. If you were at my committee meeting a month ago in person, I brought in the pennycress plant. One was tiller and one was non-tiller.
I was on.
You could go.
Oh, I covered this.
I can
Sorry,
answer
didn't
you
wanna
that
say
question.
anything, 'cause I'm not supposed to talk about that.
And they tiller so much. They have like 6 to 10 different tillers around it versus a single
All right,
stalk
okay.
plant.
So
Okay.
how far away from focal plant do these pillars emerge?
They're all connected at the base at ground level,
Oh, so
but
They're
the
not- they're
more
not-
tillers,
they're not.
no, but they could lodge there or not.
They're actually not tillers, they're really adventitious buds that are coming up from the crown area.
Tillers are reserved for grass species.
Yeah, I think
Kinda
so.
like it, but
Yeah,
that's
I
what I'm
think
asking,
so.
but-
That's what
I think that's
Raton called
Alan's
them.
getting at,
Who calls them?
yeah.
Raton, tiller versus non-tiller varieties.
So maybe he doesn't.
I have full confidence in Raton. Technicality.
Right. I mean, I think that's why you're thinking tillers. Tillers is a grass concept,
Yeah,
you know, and so it comes up like a.
yeah,
Right.
it comes from the crown.
Under,
These have a crown.
they come from that region. I don't know if
I would
you technically
go
call it a
dissect
crown.
the plant.
What?
I would dissect the plant, and see what it looks like. You don't see that there.
You might have to get a dissect, right? If you've got a big enough plant, you can kind of dig it up and see where they're coming from.
Well, that's true. Yeah, I get
You know,
that.
unless he really wants to like start getting into it. This is fun
Do they
for
come
him.
from half a centimeter above the soil or a centimeter below the soil?
So just a couple of it.
Yeah.
Well,
I
that's-
think that, this is just kind of like, thinking about this topic here. The way I view this, and I think that most people in the church are view this as well, and anyone that's a farmer in the state, soybeans is the primary crop.
Yes.
Pennycress is kind of data. It's the way you present it, kind of presented it the opposite way. Your slide was, where does soybean fit within pennycress?
Oh,
So-
you think it'd be the other way around?
I think it should be the other way around, because, and then with that, in that respect, you should move all that discussion of soybean up to the very start of the
Mhm.
time. So you actually, you might even want to introduce soybean to Canada. And let's see, how can you deal with this problem we have of the lack of coverage of soil in the system? And then talk about pennycress as a potential crop that can fit into this system.
I like that a lot. I will say a lot of these sites were recycled from my talks at the I prefer annual meeting with Pennycress, so that's probably why I was much more Pennycress focused, but
I don't think you just for the future.
Yeah.
You should use centimeters instead of inches. You said something about 4.7, almost
Yeah.
5 inches, just use centimeters.
Okay.
Get used to it.
keep it.
And don't ever revert back to inches or feet or yards
Or bushels per acre.
Yeah. Which is, yeah.
And then let's see. So the other thing, I thought I said this one time with Friday, but I don't think I said it before. And your results slides, it makes, and this is probably for everyone, the results slides, you should have a title on them that basically gives the result that you're trying to get. Some short word.
So you don't want to
Instead of
topic.
saying results, you just basically say no differences in quality traits
Okay.
in intercropping system. Something like that would be, then people can read that.
Yeah.
It's 5-10 words. They don't have to listen to everything you say, or they can if they want, but
But
they know what.
I only said that result once. And if they missed it, they missed it.
Yeah, so I mean, I think that if you think about it that way, it's, then it makes your slides easier to understand.
Okay, Gary, you mean adding the statement within the slide?
No, the title. That's
Replace the title.
The title of the slide, I think, if you have a result that's really clear, if you don't have a result that's clear, then you don't really do that. So like this one right here, agronomic trace, you could just, I mean, that would be a difficult one.
There's a lot of info here.
There's a lot of stuff there. You might be able to summarize that by saying,
I don't think, that's pretty
I
clear.
think that's pretty obvious. But that was what this slide showed, other than this other traits.
No, what it showed is that he's got one unit that's not too bad.
Yeah, that's true.
That's what this slide. shows.
Or maybe you say
Not
that.
gonna mind.
Maybe you say, maybe that's the title. Yes, whatever that is.
1146.
1146 is the idiotype that might work well. This is a type of something like that.
Yeah, something's
It's a
going on.
promising idiotype. BS 1146 is promising as a word quality for intercropping.
I mean, it gets to the point nicely.
So then it's really obvious. Oh, I know what I'm looking for.
That's another question.
Yeah.
Might interrupt you. No, Hussein, go Hussein.
I don't have a question, so more like a, I personally use Windows, right?
Oh, you can see my
Yeah.
setting up there.
Yeah.
I don't know how to fix that. It's been there for 2 years.
No, that's okay. I mean, I use both and I like both. And when I use Windows, when I export graphs, there is a specific file.
PNG, JPEG.
Yeah, something like that.
Okay.
It's the type of the file. There's a type
Yeah.
of the file that you can extract, and you don't lose any of the image quality.
Mhm.
So wherever you zoom in, it stays the same quality. So maybe you can.
No, I think that's good. I will say a lot of these graphs came from R. I just did export plot, copy plot.
Yeah. I used to do that, and then I found out about this.
Save it as an image.
Exactly.
Yeah.
If I get so fast, that wasn't.
Yeah, thank you.
So I was wondering about, we talked about this in the dry run,
Mhm.
and I thought about it a little more, so I think I brought it up in your, when I had COVID. So I'm wondering about, you said Seneca is expensive, right? But I'm wondering, would it be worthwhile to actually run an experiment with Senegrin that you buy, get a sense of what's the concentration that's required to get the phenotypes you're getting from grinding up seed?
Yeah.
And maybe it's gonna be low enough to use it, and then you can actually just buy, $1,000 worth and run the whole experiment.
Because like
don't
I
know
have,
if that's the case or not, but then you would get a sense of what's the physiological level of this compound that's needed to
elicit a response.
elicit the response.
I think that's a good idea. I have no idea how much of Senegrin this pennycress plant is producing. Like it could be very, very small levels that have a high effect.
Talk to Adrian Hageman,
Yeah.
because he will help
He
you.
can help you.
He can help you figure out how much. is
Can you
in there.
measure it in
Yeah.
the soil underneath the
Well,
pennycress plant?
You can measure how
And
much
seed
is
But
meal.
going
you're
into the
grinding
seed
up the
meal.
seed though, right?
Yeah, okay.
So you could measure it in seed. And the other thing is, the
Yeah.
reason I don't really like the seed experiment, I know you told me.
No. I
The
want,
reason I don't like that experiment is because every time you grow a batch of pennycress, and maybe not even you, because you got enough seed for your whole experiments, whatever. But the next person that comes along and wants to run similar experiments, they're gonna be growing different set of.
it's probably
And it's gonna have
environmentally.
a different amount of Senegrin, so the results
Yeah.
are gonna be slightly different, it's gonna be.
Okay.
So we just figured out how much stuff we just add.
I wish I could. That's great.
It would actually be a
I
lot
know.
easier, because then you could just mix up a big batch, take it down there and water the plants with it, and you'd be done with it, right?
Yards, they're like-
I have a horticulture minor. I'm diverse.
Can I ask a really unrelated question?
What?
What pollinators visit pennycress?
Ooh, good question. So
It was like your first slide.
Yeah.
It's like, wow, ecosystem services, it's great for pollinators. I'm like, which ones?
So I'm going to say a lot of this information I took secondhand from the I prefer meetings.
That's why you need to
There
start
are-
with soybeans. That's why you need to start with soybeans.
Yep.
Because soybeans, everybody, just say soybeans are a really important crop in Minnesota, and we really need to figure out how we're gonna cover the soil. I mean, I agree with Gary, because then I wouldn't be focusing on ecosystem services of pennycress. I thought we were going down the pennycress aisle.
Mhm.
And, yeah, you don't have,
I am firmly on the soybean side.
yeah, I wouldn't have known that from the first 10 slides.
I still think it's good to include those things.
Oh,
Something that you
I
can
agree
say,
too.
and I used to work on pollinators, is that there's a dearth of like early flowering species,
Mhm.
and so any insect which is like that's coming up, like queen bumblebees, for example, are going to come up earlier in the season.
Mhm.
Like resources are limited all year round, but especially early in the year, and so you can say it's like an early flowering
Mhm.
species, and so we'll provide like more of an extra resource
For those species, those
for
early
whatever,
species.
yeah, you have like, I don't know what species. So whoever something that is like
Very well said.
Generalist pollinators, so I do don't love it, but there are also something that could be something that is also slow to or in wild.
I bet it's flies, but we don't have to go there.
I have a
I agree
picture
with Alex, it's
of
something
a
that we
honeybee.
really need to talk about ecosystem services. And, and one of the benefits of ecosystem service is that it has something flowering.
Mhm.
Those of us from the horticultural standpoint, like, we despise dandelions because they bloom at exactly the same time as the apples bloom.
Mhm.
And they're so much easier to
Competition.
pollinate that, and get pollen from that. Basically, they go through and run the mowers to make sure that they aren't